The Knapp and Knowle cluster is a sprawling stretch of local terrain that recently underwent a major ecological health assessment. To evaluate the health of an environment like this, scientists use standardized biodiversity surveys to mathematically quantify the life within its borders. This chart shows the comprehensive survey findings. It categorizes classes of life, logging the number of species present and calculating their percentage. In the far right column, most register below 50%. For mollusks and reptiles, the survey recorded 0. Assess the ecosystem through the lens of this spreadsheet alone, and the data suggests an area that is biologically struggling. This survey tracks diversity, a literal count of unique types. It ignores abundance, the physical volume of individual plants and animals that actually occupy the land. The physical landscape of the cluster is overwhelmingly dominated by semi-improved grassland. While this habitat supports a limited variety of plants, it produces a high density of a few select species, particularly oxeye daisy and common knapweed. The fields are dominated by umbellifer flora. These are plants like wild carrot and hogweed, concentrating hundreds of tiny blossoms into a single umbrella-shaped structure. This concentration of flowers creates a massive, localized nectar source that attracts high populations of specialist feeding insects. Because these open flower structures are less accessible to bees or butterflies, the fields become a primary feeding ground for Diptera, the true flies, and Coleoptera, the beetles. The site's primary engine of life is driven by this concentrated volume of a few specialist species, rather than a broad spread of unique ones. The non-grassland species found on the survey survive by relying on a separate network of highly specialized microhabitats. The first of these sanctuaries are the 3 freshwater ponds hidden within the network. These isolated pools account for the 100% amphibian presence recorded on the survey, as well as specific pond-dwelling insects like the broad-bodied chaser and common darter. Away from the ponds, the local cemetery offers a different environment. It provides the shady, damp conditions necessary for species that cannot survive in the sun-baked meadows. This diagram highlights the difference in moisture retention. Cryptogams, organisms like fungi, mosses, and lichens, require these shaded zones to prevent desiccation. The older, undisturbed gravestones provide the stable, long-term anchor points these lichens and mosses need to establish themselves over several decades. The wider diversity of this ecosystem depends on these small environmental pockets operating alongside the larger grasslands. Even with these habitats accounted for, the low mammal and bird numbers on the spreadsheet require a closer look at how the data was collected. Researchers used an afternoon transect walk, traveling a single path during daylight hours and recording only what was visible from that route. This method cannot register nocturnal animals like bats or birds that primarily use territorial calls in the early morning. It also misses small mammals that remain hidden within the dense cover of the long grass or the scrubby hedgerows. Human observation creates unavoidable gaps in data, meaning the actual population density likely exceeds what these daylight surveys suggest. Evaluating ecological health requires looking past the raw diversity checklist. The network functions through structural synergy. Large sites provide the heavy volume of baseline life, while small sites like the cemetery and ponds provide the necessary niche sanctuaries. If the large volume grasslands are removed, the base food web collapses. If the niche sites are removed, the specialized species vanish. The health of this land depends on the interaction between high-volume habitats and these specific hidden pockets, a reality that the raw figures on a spreadsheet can only partially describe.